So let's look into the pipeline for assembly. So now we are starting with those short reads. We are talking about libraries. We get those reads, and let's look into on computers what we can do. So that thing is I define it as the pipeline, assembly pipeline. So what we do first is we do quality screening, then we do overlapping, unit tagging, where you will have unique contigs, and then we will do scaffolding, and in the end we will resolve or repeats, and our assemblies will be refined. So in quality screening, it is done while satisfying the following matrices. Number one is we look into different genome properties. For example, we look into a very important and interesting task is to look into the proportion of GC contents. Then we look into the library qualities, how good are our libraries, and we also look into the sequencing run quality. So this is about and the read quality. So this is about the quality of the data, those short read data. So first matrix is where we compare those genome properties in terms of the GC contents. So we see that this GC content, as in our eukaryotes, so we have seen in our human genome project lecture that around 40 to 45% of the GC content is there in the human genome. So if we look into the eukaryotic genome, so roughly it's around around 45, or we can say around 50% of the genome is made up of GCs. Do we see this similarity in our short reads? If it is close to that or approximately close to that, we can select that. So this is the first graph where we see that quality is good here compared to this one where the quality is bad. So we see that GC content is obviously the distribution is not perfectly normal distribution as in the previous slide, and we can see there are two mods. So we are observing two peaks here. So this kind of graph tells us that the quality may be, the reads might not be good or appropriate to use in an assembly project. Here we see the library qualities. So how good our library preps were. There is another matrix. What we do here is we take that something which we call it as 20-mer uniqueness. So we take those reads and we look into the small segments of 20 nucleotides. So in these 20 nucleotides, we are looking for how many unique fragments or how many unique these 20 mers are. So if we plot the normal genomes, which are the good qualities selected read data, we see the distribution looks like this. So as we increase the number of reads sampled, the uniqueness comes down or it decreases. So this is what is an example of where we can have a good quality data versus this one. So here we have for example, actually we are looking this into two different lines. So we have starting regions, so starting 20 mers, and we have random 20 mers. For both of them we see a different picture here. So this is the example where we have our number of percentages of the unique stays at 0.2 or 20%, even if we keep on increasing our number of read samples. So it's not touching the line here as it did in the previous case. So that might not be an appropriate data for an assembly project. Here we see the run quality, sequencing runs, how they are good, and does sequencer give us the good data. We are looking this in terms of the number of base counts by read position. So we know that we have A, T, G, and C, and sometimes sequencers places N where it's unknown or where it's not sure which nucleotide is there. So we have these different colors representing those different nucleotides. So if we place all of them together, we can have something like this here in this line. So it's basically 2e raised to power 6, so slightly over 2 million. That is for each position, number of these bases. And if we want to look them individually, all these four colors, they merge here. So we have around, for example, 500, roughly around 500,000 each of them. So this is the example where we can have a good run quality compared to this one where we have the bad quality. So we see up there the trend is similar, but here we can see a huge variation among the composition of different nucleotides. So ideally it should be these nucleotides, they should be close to one another. There is one more thing, that G's should be corresponding to C's, and A's should be corresponding to T's, because in the DNA they pair up with one another. So as many A's, as many T's, as many G's, as many C's, you must see them. But here if you look into this picture, there is some huge difference among these proportion of these different nucleotides. So this may not be a good data. Here is another matrix. So that is the read qualities. So we are comparing the quality of those base calls at each positions, and we have the quality scores, the average quality scores, and on the x-axis you have read position 0 to 80, and here we have the scores. So at each position we stack all the reads together. Actually we stack their quality scores and we see what we have in terms of their median scores right here. So if we see, we start from the left hand side from nucleotide number 1, we see the qualities are really good, close to 40, and then it keeps on decreasing, but that is quite expected in Illumina, that towards the 3' end of those reads or the end of those reads the qualities are slightly low, but they are still higher than threshold. Normally in our experiments we have the threshold which is 20, so any score above 20 is good. So this is the example where we can have a good quality data compared to this one where we have a bad quality data. So in the beginning the qualities are really good, but somewhere in the middle it break off, so the qualities they go really low. So obviously this example is where the data has not passed through this quality filter. So screening is done while satisfying these matrices, and unless most of these matrices are not satisfied, we cannot start this really sensitive procedure. You know, this assembly is sensitive because after you perform an assembly and after that assembly is submitted, people will come and compare their sequences against those assemblies. So in order to have a really good assembly, we must have a really high quality data.